Hi there, in this video we're going to look at how things pass across a membrane. So as we know, our membrane is a phospholipid bilayer. In this video I'm just going to draw it as two lines like this representing the two layers of phospholipids. So the first way that things move across the membrane is by simple diffusion. Now in simple diffusion, molecules move from an area of high concentration to an area of low concentration across the membrane without the use of ATP. So they go from high concentration to low concentration. Now, because they're going high to low, there's no ATP required, and they're literally just going through the phospholipids. Okay. The type of molecules that can do this are small molecules, molecules that are non-polar, and molecules with no charge. Okay, they can literally, they're tiny little non-charged molecules. They just squeeze in between the phospholipids and diffuse across from high to low concentration. Now, some molecules can move by diffusion, but not as easily as simple diffusion. And these ones use something called facilitated diffusion. Now, the word facilitated just means helped. So at the moment, I'm helping you revise. I'm facilitating your revision because it's easier, isn't it, to listen to me explain it than have to decode it from a book, to read it in a book. So it just makes it a bit easier. It makes it easier because you've got images, etc. So these molecules are still going to go from a high concentration to a low concentration because it's still diffusion. They're still not going to use any ATP because it's still going down the concentration gradient, but they need a little bit of help. And the help is either in the form of a channel protein in the phospholipid bilayer or in the form of what we call a carrier protein in the phospholipid bilayer. So the molecules can simply go through this channel and out the other side, or they can go into this carrier. The carrier swaps shape and it comes out the other side. Okay, so they have to go through proteins, and there's two types of proteins that they can go through. The types of molecules that can do this are the ones that can't do simple diffusion. So larger molecules, polar molecules, and charged molecules. Sorry, I'll write charged over here. So if they can't go by simple diffusion, but they still need to go from high concentration to low concentration, they'll go by facilitated diffusion. Now, a third type of diffusion is osmosis. Now, osmosis is simply diffusion of water. It's still diffusion, which means it's still going to go from a high concentration to a low concentration across the membrane. But the word osmosis simply means diffusion of water molecules. So we specifically got water molecules that are moving from high concentration to low concentration. Now, when we're using water as our example, you can also say that it goes from high water potential, which has a symbol like this, to low water potential. So water potential can be written simply as that symbol. Okay, so you can either say osmosis, it goes from high water concentration to low water concentration, or high water potential to low water potential. And the type of molecules that can move across in that way are only water molecules. Okay, it's not about size, polarity, or charge. It's just water molecules, and there's no ATP. Okay, it's still diffusion, still going from high to low concentration, no ATP. Now, the other type of method that things can move across the membrane is active transport. Now, active transport occurs if things need to go up the concentration gradient. So that means they're going from where there's a low concentration to where there's a higher concentration. So it's like they're going in the opposite direction. All right, so here's our membrane, and they want to go from low to high concentration. They cannot go by diffusion, facilitated diffusion, or osmosis because they're all high to low concentration. These molecules want to go low to high. Now they have to go through a carrier protein. They can't use a channel protein. It has to be here a carrier protein. So the carrier protein is an intrinsic protein. It goes right the way through the membrane, and the molecule simply binds on this side. The carrier protein changes shape, and the molecule is released out the other side. So it goes low to high concentration, and this one does use ATP. Because it's going the wrong direction, it's like it's going up a slide. It's really hard to go from low to high concentration. So it needs ATP to be pumped across the membrane. Okay, and these little proteins, remember, are called carrier proteins that enable it to cross the membrane, basically in the wrong direction. Now, the last process is bulk transport. And what happens in bulk transport is we're able to transport molecules that are too big, or there's too many of them to be able to be transported by facilitated diffusion or simple diffusion or whatever. So if we want the molecules to leave the cell, the process is called exocytosis. And what happens in exocytosis is that the molecules that we want to get out are inside a vesicle, and that vesicle moves to the edge of the cell and fuses with the cell surface membrane. And when it does that, eventually it opens up and the contents is released. And it's very important in our descriptions that we talk about how the vesicle fuses with the membrane. Now, the opposite process to that is called endocytosis. So exo is like exit, endo is like enter. So molecules going in. So endocytosis is when molecules move into the cell. So if this is our cell, the molecules are next to the cell, and the cell starts to form a different shape to its membrane. It sort of tries to engulf it, it surrounds it, and eventually it forms a vesicle around it, and the molecules that are taken in are enclosed in a vesicle inside the cell. So we say that it engulfs. Okay, so it engulfs the molecules in order to take them in. You need to mention in endocytosis as well that when it's taken in, it's then inside a vesicle in the cell. So we must mention the word vesicle in our answer. Okay, so exocytosis releases lots of molecules, and endocytosis takes these molecules in. Now, there's a very famous example of endocytosis, which is done by phagocytes. Now, phagocytes are cells in our immune system that engulf pathogens. So these little molecules here might be bacteria that have infected us, and a phagocyte engulfs the bacteria and then uses enzymes to digest them to get rid of them from your body. And that's a really, really famous example of endocytosis that you might have come across before. And that's the end of that video.